Alrighty, well, hello everyone. Professor Kyle back again one more time for a recorded lecture. This will be the last in this video series and probably the last you guys will ever hear from me ever again. I'm not dying. I'm just not going to be your professor anymore, so you guys won't have a reason to watch any of my videos. Terribly sad, I know. But with that being said, today we're going to be covering eyes and ears, all right? Our special senses, our auditory and visual senses. As you can tell, the caffeine is kicking in since we previously spoke. So hopefully I will be even faster this time. But again, as I mentioned previously, I'm not going to go on as many tangents. I can't promise that I won't go on any, but I'm not going to go on as many side tangents discussing physio and that sort of stuff in this lecture as I normally would in class, just for the sake of keeping it nice and concise and focus in on the information you need to know for your exam. Again, this video is specifically for my own students. Alrighty, with that being said, let's go ahead and hop on in. That was anticlimactic because I didn't have PowerPoint selected. All right. So looking at our ears, we're going to start off with auditory. All right, so let's talk about our external acoustic. Wow, the video is lagging. All right, we're caught up. We're going to focus in on our external ear first, which is this environment over here. Starting things off, we have this term called the external acoustic meatus. Hopefully that sounds familiar to you. Previously, we discussed it when we did our cranial bones and such. And in that framework, we specifically were referring to this space right here as the external acoustic meatus. Although now that we've kind of zoomed out and we're including our other anatomical structures, technically the external acoustic meatus is this entire tube right here, okay? Now, with regards to the ear itself, we have three features that we are going to want to know. We have the auricle, which is kind of like the plate surface of the ear. Obviously, I have headphones on, so you can't really notice it, but like it's this large plate portion. Then we have the helix. Okay, that is what you think of as kind of like the outer edge or crest of your ear, if you will. And then we have what we commonly refer to as the lobe of the ear. Okay, your ear lobe. Technically, it's though we should call it the lobule. Okay, so don't forget that U. It might sneak up on you on a test, but this is what you most typically would get pierced. I usually ask this question in class. I can't in this format. Sorry, my throat's a bit scratchy from talking for so much. But the ear lobe itself is composed of the external ear is composed of elastic cartilage. That's normally a question I ask in class, but I can't hear. That's basically all we need to know with the external ear. Congratulations. We're going to go ahead and focus in on some other structures which are noted here on the side, but we have slides dedicated to them, so we're going to go ahead and zoom on in. Specifically, we're zooming in on the middle ear, okay? So this segment right here, the middle ear environment, that is going to be this slide right here. All right. Now, first and foremost, we have three of the smallest and arguably coolest bones in the human body. They are called the auditory ossicles. All right, there's these guys right here. Please remember that term and remember these three bones. We'll take each of these in order. On the next slide, we'll talk about a little memory tool for each of them. But I just want to pass something out for you guys of how sound and our perception of it works. So I use this example in class. You're dealing with it again. Congratulations. Or if you're not one of my students and you're watching this video, welcome. All right. So imagine someone is serenading you or you are serenading yourself playing on a guitar. All right. So when you play a guitar string, the camera will grab it. All right. We then cause a wave oscillation in that string. Those wave oscillations will cause an impedance within the air environment. Those impedances will eventually make their way through the air and eventually interact with our tympanic membrane, otherwise known as our eardrum, right? Our eardrum will absorb those signals and then transmit them through a sequence of these three bones, which are the malleus, the first one, shaped somewhat like a mallet, hence its name. The incus, which is shaped kind of like an anvil. If you ignore this whole long projection down here, it kind of looks like an anvil up there. And then transmitting that to the stapes. From the stapes, it will then be shunted into the cochlea. So on our last lecture, we talked about the vestibulocochlear nerve. Okay, this guy over here, this is where that name comes from, right? So we have the cochlear portion of the vestibulocochlear nerve, and we have the vestibular portion. The vestibule associated with your sense of equilibrium, cochlea associated with your sense of hearing. Circling back to our example, sound waves have hit our tympanic membrane, are transmitted through this kind of ratcheting mechanism, and compressed down into the smaller cross-sectional surface area at the back of the stapes and then shunted into the cochlea. There's technically 3 tubes in the topia. We're not going to worry about the precise anatomy and physio of our hearing. But do note that one of those tubes contains hair-like projections, and those hair-like projections are varying sizes, and each of which has its own individual nerve. So if that hair gets activated because it vibrates with the particular frequency of sound that is being transmitted, then that signal will travel down its own unique nerve into the cochlear portion of the nerve, back into the brain. And your brain, based off of which nerves are getting activated and their associated hairs, is able to then determine the pitch or the tone of the sound that is being heard. The way that works in terms of the distribution of sound perception within this space is you have shorter hairs and therefore perception of higher frequency sounds in the proximal portion of the cochlear tube. And then we have longer hairs perceiving slower or longer wavelength sounds in the more distal portion of the tube. Human hearing typically is between 20 to 20,000 hertz. So 20 hertz means 20 oscillations per second to 20,000 oscillations per second, right? So pretty decent range, although other organisms have much better. Why do we have this sort of multi-stage process right here that we just described? Well, it does a couple different things. The tympanic membrane is doing 2 things. Not only is it responsible for our perception of sound. Please tell me I'm recording. I am. Cool. But it is also protecting this very sensitive environment of our middle ear from the external environment, because generally speaking, this should be a sealed structure, right? So we're not getting a lot of air containing pathogens and that sort of stuff into this very sensitive structure that would be prone to infection and therefore inflammation that would compromise our hearing. Secondarily, the tympanic membrane, although it's small, it's a relatively large cross-sectional area, and I should note that the general function of your earlobe into the external acoustic meatus is to essentially basically shunt sound and focus sound into a much smaller space, if that makes sense, right? Okay. Other organisms that are more reliant on their hearing have much larger ears. If you think like a fox or a bat, something like that. Again, we're primarily visual, which we'll talk about in a bit. But that eventually hits this fairly large cross-sectional area, and then that sound, by getting transmitted through these auditory ossicles, is then compressed into a much smaller cross-sectional area over here before it then travels into the tube. This essentially acts as a sort of sound amplification system as well, so it's enhancing the sensitivity of our hearing. Another structure that we should certainly note is this guy right here, the pharyngeotympanic tube. Okay, pharyngeotympanic tube. It's also known as the auditory tube or the auditory canal or the eustachian tube. To me, those are all acceptable names. What is the function of this tube? Well, this tube runs from the middle ear environment that we see right here down actually into the back of your sinuses, okay? And the primary function of it is to maintain equilibrium in terms of pressure between the middle ear and the external ear environment. Your hearing becomes compromised if there's a different level of atmospheric pressure within this space versus within the external space. The one example I'll give, again, this is the slide where you got to give me some room for tangents, is you can imagine if you have a sinus infection, okay? Man, the video is really lagging. I'm sorry. There's a brief pause. I don't know what's going on with my camera. We're just going to roll with it. I apologize for any sort of delay in the video feed. I believe we were talking about pharyngeotympanic tube. All right. Get my laser pointer back. There we go. So it runs to the back of the sinuses, all right? So if you can imagine if you have a severe sinus infection, that sinus infection will lead to inflammation. Oftentimes that inflammation will close off, as well as the congestion itself of the mucus production, will close off this tube, and it'll end up being that you have a muffled hearing, and you might also sense a sort of echoiness in your hearing. That's due to the congestion kind of closing off the nasal environment. That's also due to this tube getting closed off, meaning that the pressure cannot be equalized within your inner ear, or sorry, within your middle ear and the external environment. This will lead to a muffling of your hearing. Also, when you take off in a plane and you know your ears pop, that sort of thing, that's this eustachian tube equalizing the pressure because the pressure in the external environment changed, and then that restores your hearing. All right, enough tangents there. The vestibule, kind of similar to the cochlea, it is lined with hair-like projections within these tubes. There are three vestibular tubes, each of which have a bit of a fluid and hair-like projections. Your brain is able to detect the shifting of fluid within those tubes and its activation, its interaction with the hair-like projections to essentially determine where up and down are, as well as your sense of momentum. Okay. I apologize for that Windows notification. Yeah, I won't go on more tangents than that. We'll move forward. All right. So little kind of visual memory tools for our auditory ossicles right here. So we have the malleus, shaped something like a mallet, right? This guy right here kind of looks like a mallet. This, the incus anvil, a little bit more of a stretch. It kind of, if I just look at this part, it kind of looks like an anvil. So we'll go with that. And then the stapes very much does so look like a stirrup, okay? Anything else we really need to note here? I think that's basically it. Alrighty. Now, that's all we need to cover for auditory. I would love to talk about it more, but we will leave it at that. Now we're going to talk about the eye. So first things first, we're going to talk about our motor nerves associated with the eye. We discussed this previously when we were covering cranial nerves. So here I have them labeled out as to each muscle and its associated cranial nerve that is controlling its function. In terms of naming conventions and physiological function, which you need to know, so you need to know the name, cranial nerve associated with it, as well as the action of like what it's doing to the eye, how it is moving the eye. Don't be too intimidated by this. It's relatively straightforward, okay? Now, what we know about muscles. Well, we know that muscles, when they contract, they decrease their length, right? Their longitudinal area. So this muscle will contract, it will contract and pull the eye this way, right? Because this tissue is decreasing its lengthwise area, those myosin and actin filaments ratcheting against each other. Knowing that bit of physio, 4 of these muscles will now become immediately very easy to remember, okay? So my general rule is whatever the orientation of those muscle fibers are and where their point of attachment is on the eye, and this also applies to the rest of your muscles as well, your skeletal muscles, is going to be the direction it's going to pull the eye. So in the example that I've kind of illustrated here with the arrows, we have the lateral rectus muscle, okay? So this is looking from, I think your guys' wait, let me turn this way. Okay, we're looking this way at the person's surface. So their cheek would be like right here, right? This isn't like a mid-sagittal to the side. This is the right eye that we're looking at. So therefore, this is the, for me, I know the camera's flipping. This is the right side of my face. So on the outside edge would be the lateral muscle because it's on the lateral side. Makes sense. All right. So if that muscle contracts, it's going to pull my eye laterally. It's going to pull it outwards. All right. That applies to the superior rectus, which the name got cut off right there, the lateral rectus, the inferior rectus, and the medial rectus. Now, just to tie this together as an example, so the camera is over here in the corner. All right. If I want to look at that for my right eye, actually, no, let's cover left eye. For my left eye, my lateral rectus muscle is going to contract, and that is going to pull my eye towards the direction of the camera. For my right eye, it's not going to be my lateral muscle because that's on the wrong side. It's going to be the medial rectus muscle that's going to contract right on that middle surface. That is going to contract, and that is going to pull my right eye to look at the camera. Hi, how's it going? So, knowing that, that's 4 of our 6 immediately covered. And then we have our 2 unique muscles that have their own dedicated cranial nerve associated with their function. And they also have kind of a unique orientation, affecting how altering how they move the eye. Let's start off with what I believe to be the coolest one, which is the superior oblique muscle. So as we can see, the muscle belly is relatively parallel with the superior rectus muscle initially. But then it does this weird looping thing, okay? It runs through this little bit of connective tissue known as the trochlea. We've seen that term before, trochlea. And then it comes across and inserts onto the eye on the lateral surface of the eye. Whoa, okay. What does that mean for the movement of the eye? Well, since it's kind of running along this simple pulley mechanism, if this contracts, okay, if this pulls back, that's going to pull the eye in 2 directions. Going to do 2 things to the eye. So if this moves this way, then that's going to pull the eye from the lateral surface this way, okay, which is going to lead to a downward tilt in the eye. And since it's on the lateral surface and we're pulling that, the eye is also going to drift and look laterally. Okay, so superior oblique muscle, it depresses the eye because, again, this distance is getting reduced, which is going to pull the eye downward. Sorry, there's lots of lines getting added. And it's going to turn it laterally, right? It's going to turn it laterally because the attachment is to the lateral surface of the eye. So it's going to pull the eye kind of outwards like that. Okay, similar concept with the inferior oblique muscle. Okay, except there isn't a pulley mechanism. It's just the orientation of the muscle itself is different. So here we can see we have an origin on kind of the medial surface, middle surface of the eye, and then attachment on the lateral surface, but it's wrapping under, okay? Wrapping under the eye. What that then means, so inferior oblique being right here, is this is going to elevate the eye because one direction of this pulling mechanism is going, I'm going to use my color coordination here again. So since we have attachment here, one direction of the pull is like that, okay? So that's going to pull the eye up. It pulls the eye up. And then also it's going to turn it laterally because, again, that attachment surface to the lateral side of the eye. So it will pull it this way as well. Does that make sense? Hopefully it does. This is the best I can do with a static image. All right. That's basically it. That's all you need to know for the muscles. Now we're going to get into the more fun stuff with the general anatomy of the eye. There's a tiny gnat in my room, and it's really annoying. All right. So to keep this relatively simple and move relatively quickly, we are just going to follow the path of the eye, or path of light through the eye and the structures that it will encounter in sequence. So first things first, when light comes in, it's coming from this side, okay? First thing it's going to interact with is the cornea. That is the most superficial portion of your eye lens. It is upon which you would place like a contact lens if you use those, okay? So light passes through the cornea. That begins the process of focusing light towards the back of the eye, where eventually we're trying to get all light into focus by the time it hits the retina. Then it's going to pass through a liquidy substance known as the aqueous humor, okay, produced by the ciliary bodies up over here. And then once it passes through that liquidy substance, it passes through an open space called the pupil. Okay, so the pupil is the empty space formed by the iris. The iris is what you think of with the color of your eye. Whether you have blue eyes, brown, green, hazel, that sort of stuff. So the iris is the smooth muscle that is essentially regulating. It's acting like an aperture if you're into cameras, regulating the amount of light that passes through the pupil itself, which is the open space. Then light interacts with the lens of the eye. Then it passes through vitreous humor, which is a sort of gel-like substance. It kind of gives general structure to the posterior chamber of the eye. And then lastly, it'll finally interact with the retina, which is where your photoreceptive cells are. Here we're looking at the eye. This is kind of the inside surface of the eye. So like this is so the perspective would be kind of looking back out of the eye, if that makes sense. All right. So we have the entirety of these tissues are known as the ciliary body, okay? So this whole chunk. The portion of the ciliary body that you can see is actually your iris, okay? So it's a portion of smooth muscle. So that would be this little bit right here, kind of more of a yellowy orange color. Then we have ciliary processes and suspensory ligaments that are attaching then to the lens of the eye. So the ciliary muscles, part of their job is regulating the amount of light that comes into the eye through the iris. The other part is they're actually associated with helping pull and apply tension to the lens of the eye to adjust its shape. The more stretched out, the harder they pull, the more narrow the lens of the eye is, and therefore the more straight light will pass through it. And then if they relax the shape of if the muscles relax, then the lens will return to a more neutral shape, which is a more curved shape, in which case it will bend light more significantly. It's all part of that focusing process of the eye. And then again, anything else we need to know here from that perspective? No, those are the things I wanted to point out there. Let's focus in on the back of the eye. So the yellow layer, the most superficial, this is the photoreceptive layer. This is your retina, okay? So this is where we actually have perception of light passing through our eyes. The fovea centralis, this is what we think of as the center of your vision, okay? So when you imagine you're looking at something and you're focusing on something, well, what you're focusing your eyes to is so that the light of bouncing off of that object is hitting the fovea centralis. So this would be the center point of your vision. The other structure we need to know on in terms of the retina itself is this. This is known as the optic disc, okay? So the most superficial layer of the retina, which we'll talk about in a moment, is actually the neural tissue itself. Light has to pass through the neural layer to get to the photoreceptive cells just underneath. And the axons of all of those neural cells are running along the surface of the retina, and then eventually they are shunting backwards here to form the optic nerve, okay? What that means in practical terms is due to that structure, there is no room for photoreceptive cells in this area because it's all just the neural cells, the axons of the neural cells running backwards to form the optic nerve. The net result of that is, since there are no photoreceptive cells, this is actually a blind spot in your vision. Now, you might be saying, hey, Kyle, well, I don't see a blind spot in my vision. Maybe I don't have it, or maybe it's so small I can't notice it. No, it's there. For me, if I look straight at the camera, and I should be able to do this. Obviously, you don't have depth of perception here, but there's a blind spot in my vision if I look at the lens of the camera right about here in my vision. Small little dots. Relatively small little dots. Your brain is very cool. Your brain has adapted to this limitation in the structure of the eye. And so what it does is it is constantly filling in those gaps, those little black dots in your vision, with whatever it thinks most likely should be there based off of context. You can actually look up ways to find the blind spot in your eye. The way I do it is I just take like a sheet of paper and you can put an X on, you would put like an X on one part and then a dot on the other end of the page. And then you look at the dot with one eye, and eventually you will find a spot where the dot will disappear, and that is the blind spot. And at that point, it'll look white because your eye is using context cues of, hey, I see all of this white stuff around the blind spot in my vision. And so your brain just basically fills in that dot with white because that's its best guess as to what is there. Fun stuff. Anything else we need to talk about? Yes, two other layers here. So yellow is the most superficial layer. This is the retinal layer. Behind that is the choroid. This is the vascularized layer. Okay, this is where all the blood vessels and stuff supplying nutrients to the highly metabolically active retinal cells exists. And then behind that, we have the sclera, which is actually a form of connective tissue. And this is going to be the outermost shell of the eye. This is what is giving it its general shape. All right, so here we just have a brief slide summarizing essentially that pathway of light through the eye. So first thing it interacts with is the cornea, that most superficial layer. Then it passes through aqueous humor, relatively liquidy substance produced by the ciliary bodies. Then it passes through the open space of the pupil. It's not a structure itself, it's just a gap. Then it hits the lens. The lens further focuses the eye. Again, it's attached on suspensory ligaments to portions of the ciliary processes, which are applying tension and helping adjust the shape of the lens to focus light. Then it passes through the vitreous humor, this sort of gel-like substance back here, filling the posterior chamber of the eye. And then we have the retina itself, which is where the photoreceptive cells are. Speaking of retina, let's talk about it. So within the retinal layer, we have 4 different types of cells that we need to know. We have our photoreceptive cells that we've been referencing at length. Those are our rods and our cones. We'll talk about those in a little bit. Then we have our bipolar cells. Our bipolar cells are essentially doing this job of mediating the signal from our photoreceptive cells and then kind of consolidating it back to our neural ganglion cells. We'll talk about that process of, it's known as convergence, and we'll talk about why that's important in just a moment. And then we have the ganglion cells themselves, right? All right, so these are the guys that are running back to the optic disc and then forming the optic nerve, going back into your brain, allowing you to perceive, provide our sense of vision, okay? Now, let's break things down a little bit more. So with our photoreceptive cells, we have our rods and our cones. You've probably heard those terms before. Rods are associated with high sensitivity but lower resolution vision. Why is that the case? Well, first of all, the vision they provide is black and white. They are essentially detecting the presence or the absence of light. Therefore, the vision they provide is black and white. Conversely, we have cones. Cones are actually photoreceptive only to specific segments, specific wavelengths of light. Therefore, they allow us to have color vision, okay? Now, this is where that process of convergence and the importance of bipolar cells comes into play. In the case of rods, we see a high degree of convergence. What that means is we will actually have many individual photoreceptive rod cells, okay, many cells covering a specific portion of our vision that are all converging to the same bipolar cell. You can kind of see that. Does this work? Yes, it does. You can kind of see that right here. Okay, so I can't use the laser pointer while I'm zoomed in, but notice there's three kind of purpley blue cells converging to a single bipolar cell. The bipolar cells really look like Patrick Star from SpongeBob. I don't know why. So that's demonstrating that notion of convergence, right? All right. That process of convergence allows us to dramatically increase the amount of surface area that we are working with to interact with light, right? So this increases the sensitivity of our vision. The negative consequence of that, however, is that because we're dedicating a fairly large surface area with all of these rod cells to a single bipolar cell, that means we have one big pixel now, right? So we have plenty of opportunities to interact with photons, but we're only going to be able to give one on or off signal for that large surface area, meaning that we have a very low resolution image, okay? But in a dimly lit environment where there's not a lot of light, that's better than nothing at all, right? Then we have cones. Cones actually demonstrate very low convergence, okay? There's usually at most only a couple of cells to a single bipolar cell. And by the time you reach the fovea centralis, right, this structure that we mentioned right here, the fovea centralis, the center point of your vision, it is basically exclusively cones, and it is exclusively cones that are in a 1-to-1-to-1 relationship, meaning that there is one photoreceptive cell to one bipolar cell to one neural ganglial cell. What that means is while we have very low sensitivity in the center of our vision, because there's only one opportunity to interact with a photoreceptive cell per nerve, we also gain the benefit, though, of having very high resolution because each one of those photoreceptive cells is its own unique pixel in our vision, right? So very high resolution, lower sensitivity, okay? In terms of general distribution along the periphery of your vision, you have a higher concentration of rods and a lower concentration of cones. And then as you approach the center, the proportion of cones increases and the degree of convergence decreases, right? That's why if you're in a nighttime environment, right, and you need to see something that's in the dark, you might look slightly off center of it and then all of a sudden you'll be able to see the object a bit better. That's because that light is now interacting with more rods, which are going to be sensitive in that environment. Alrighty. Anything else we need to talk about here? No, we don't need to get into the physiology of like rhodopsin and all that sort of stuff. All right, so there are videos associated with this stuff. You can watch those on your own if you are so desire. I'm just going to quickly summarize the information, the kind of net product of watching those videos. So myopia is nearsightedness, okay? This is when the shape of the eye is too long, meaning I'm going to do a poor illustration here. All right, so if we have an eye like this, and then, this would be the cornea layer, okay? If the eye is too long, then that means light coming in is coming in. It's getting bent by the cornea and the lens, and then it ends up coming into focus at a point in front of the retina. Oh my goodness, the drawing is terrible, I know. But it's coming into focus here rather than over here on the retina itself. So by the time it hits the retina, these light rays will now be basically going out of focus. Hopefully that makes sense. I know the drawing's terrible. Conversely, hyperopia or farsightedness is when the shape of the eye is too short. So I will do another drawing right here. Okay. Make it bigger so I have more margin for error. All right. So what this means is light is coming in. It's hitting our lenses and the cornea and stuff, and it's coming into focus, but it's actually coming into focus at a point behind the retina over here, right? So it's never quite in focus by the time it gets there. All right, that's myopia and hyperopia. And then astigmatism is just a relatively undulating surface. So rather than being, smooth like this for the cornea, if there's this sort of undulation and roughness to the surface, this will cause light rays to kind of bounce off in weird directions. It causes a blurriness in your vision. So LASIK eye surgery is correcting these processes by altering the shape of the cornea. Again, that is that superficial layer out here, first thing that light interacts with. And we can alter that shape and therefore fix some of these issues. Cool. We use LASIK eye surgery. This is using small lasers to remove almost individual cell layers of the cornea in order to adjust. So if we have an astigmatism, right, we'll slice into this with lasers till eventually we get a nice smooth surface. Cool. Glaucoma is when there is excess pressure within the eye, most commonly is caused by excess production of aqueous humor. Okay, so two things that are happening here. The ciliary bodies produce the aqueous humor. If we have too much aqueous humor being produced, we will have problems of having too much pressure. Or if we're just not getting rid of it fast enough, if we're not draining it out, then eventually we'll have too much within the eye itself. This will then cause pressure to build up, which can damage the retinal, very sensitive retinal cells, okay, the neurobipolar and photoreceptive cells, and it can cut off, circulation in the choroid layer behind that, kind of infarcting the tissue, causing it to die of lack of oxygen and nutrients. And then we have a various methods of treatment for that. I'm not going to spend too much time. You can just read these on your own, trying to get this video done relatively quickly. These are all different ways to either do one of these two things, right? Which is either to decrease production, right? Decrease production of aqueous humor, or increase the drainage rate. Those are our two strategies for fixing glaucoma, and that will then reduce pressure inside of the eye. Then lastly, we have cataracts. Okay, cataracts, those are cloudiness within the vision. So the lens of the eye is predominantly composed of proteins. Over time, these can kind of form these sort of weird crystalline structures within it that will cause for light to refract in weird ways, ultimately leading to distortion and cloudiness in your vision. Okay, cloudy, blurry vision. You can, if you've ever seen like an old dog, dogs are very prone to developing cataracts in their later years, and that will be kind of a cloudiness in their eye. You can especially see it if you shine a flashlight kind of on a tangent angle into the eye. And then the example procedure that this video discusses is called phacoemulsification, which is basically you break down the lens and then you insert an artificial lens, oftentimes composed of like a silicone or some sort of polymer structure. Modern phacoemulsification and artificial lenses are usually multifocal, meaning that you can still focus at multiple distances. It used to be the case in the earlier days of these sorts of procedures that the lenses would be monofocal, meaning you'd have to essentially, as a patient, pick a particular distance that you wanted to have in vision, be in focus, and then you would need to use glasses for all other distances. But modern lenses are fairly good at being pliable. It also depends in part on the state of like the ciliary processes and suspensory ligaments as to whether or not that is viable. Then lastly, we have the dissection itself. If you are just a random passerby on the internet, this won't be relevant to you, but for my students, this is very relevant, so please pay attention. All right, so these are the structures that you are responsible for identifying during your dissection. So we have the cornea right here, okay, this little bit. And then what we're seeing right here, this layer, this is part of the sclera, okay? The white stuff over here, this is adipose tissue, ignore that. This is part of the sclera, that outside surface of the eye. So you need to be able to recognize the sclera, the cornea. Extrinsic muscles like are kind of running out over here, okay? And the optic nerve you can see right here. All right, internal structures you need to know within the anterior portion or the anterior chamber of the eye. You have the ciliary body, which is suspensory ligaments to the lens. You have the iris itself, which is what you think of with the color of your eye. We have the pupil, the open space, and the aqueous humor. When you initially perform that incision by kind of cutting into the side of the cornea like this right here, then it'll kind of squirt out a little bit of aqueous humor. Oftentimes, I would have warned you to be careful, wary of that. And then in the posterior portion of the eye, we have these structures, which actually will make more sense if I go. Well, no, it works here. Why am I zooming in? So let's just zoom in right here. Okay, so you can see the lens kind of floating on the island of the vitreous humor, okay, on the left-hand portion of the eye. On the right-hand portion of the dissected eye, this would be that second cut you would have done during the dissection. We can see the optic disc, that point of convergence. So the most superficial layer, it's kind of got a slight pinkish hue. It would have with your dissection eyes. Are all converging to a singular point. That point of convergence is the optic disc. That is the blind spot for the eye. All right, and then under that would be what's called the tapetum lucidum. So I'm going to zoom back out. It's that sort of oyster bluish green layer that we can see here. I'm in the way of it. It's okay. Organisms that are specialized in night vision or organisms that need good night vision oftentimes have this layer that our eyes do not have, called the tapetum lucidum, is essentially a reflective layer just behind the retina itself. So light comes into the eye. I'll do a quick drawing. All right, so light traveling this way hits the retina. Okay. It's the retina, passes through, and then it hits this tapetum lucidum. It hits this layer right here, the TL. All right, so it hits the tapetum lucidum, and then it'll get reflected back out. Okay, so this essentially gives us two opportunities. It gives that organism two opportunities for the photoreceptive cells to interact with a photon of light. And if you shine a light at an organism that has tapetum lucidum in their eyes, then you have that bright glowiness within it. Obviously, we don't have it, therefore we don't have that sort of reflectivity. Anything else we need to know? This is just another summary of the dissection you would have already performed. Make sure you can identify these structures. Oh my goodness, we are finally at the end. Congratulations. Yeah, we've done it. I've done it. You've done it. Great job. Wish you the best on this exam. And regardless of whether or not you ever see my face or hear my voice again, best wishes and every blessing on your future endeavors.